This video is basically just going over the basics about carbon chemistry. Some of it you will have learned in chemistry, and some of it will be new to most of you, or if not all of you. And so it's important that you understand how carbon chemistry is different than just regular inorganic chemistry. And it's foundational to a lot of things that we'll be learning about. So pay close attention. Make sure that you ask any questions that you have. Be sure and submit to me or ask me in person or whatever you need to do if you have difficulty with some of this kind of stuff. It's not difficult, but it is a little bit different because it's carbon. So carbon is unique in a lot of ways. It's very versatile and lends itself to a lot of different variations. Those 4 valence electrons allow it to make various different kinds of bonds. It can also bond with other carbon atoms with single, double, or triple bonds. It can have single, double, or triple bonds with other atoms as well. And so that's going to be important it makes large complex molecules possible because of the variations that can occur in the carbon bonding. So it's important to understand that it is unique in the way that it bonds. Are there other atoms that can bond this way? Probably, but they haven't lent themselves to making organic compounds. So that's what's important here. Carbon chains of different lengths and shapes are going to make up the skeletons of most of the organic molecules. You don't need to worry a whole lot about this. You don't need to learn the naming conventions or anything like that. At some point in the future, if you stay with biology or chemistry, biochemistry, you'll end up taking organic chemistry and learn a lot more about that kind of stuff. But you do need to know, big thing to know is that changes in structures, like something simple like the location of a double bond in the chain, or whether the molecular formula is different, whether there's a branching or something, all of those little tiny changes can actually have a major effect on the function, because in carbon molecules and carbon compounds, structure and function are closely related to each other, and so if the smallest change in structure can definitely lead to a change in the function. So pay close attention to that kind of stuff. Don't worry about the terms butane and methylpropane and that kind of stuff. You don't need to know that stuff at this point in time. Isomers have the same molecular formula but different structural formulas, and since their structures are different, like we said before, they're going to be different properties. And so it's important to understand, again, you don't have to know a lot about this. You do not need to know whether they're cis-trans isomers or enantiomers or any of that kind of stuff. That's not important to what we're learning about here. But the big thing to remember is that when the structure is different in some way, whether it's just flipped over the other direction or whether the functional groups are in slightly different positions, definitely affects the function of the molecule. And that's really important to what happens. There are a lot of different functional groups that can be attached to the basic carbon skeleton. They're called functional groups because they can affect the function of the molecule. Makes perfect sense when you think about it. The 7 most important ones are the ones that you need to know about for this class, okay? You need to know about the hydroxyl, carbonyl, carboxyl, amino, sulfhydryl, phosphate, and methyl groups. Some of that you may, again, some of these things you already know. You know a little bit about phosphate groups and hydroxyl groups or hydroxys from chemistry, but some of the others you may not know as much about. So we need to talk about those briefly. These are the basic structures of those kinds of things and the kinds of molecules. This table is in your book in Chapter 3. And you don't have to memorize it by any stretch of the imagination, but you do need to know some basics about how they work and some of the properties that they give to the molecules that they're attached to. For instance, a carbonyl group, I'm in a carboxyl group. A carboxyl group is an acid group. If you have a carboxyl group on a molecule, it's going to have acidic properties in that particular part of the molecule. An amino group is a basic group, so that part of the molecule where the amino group is attached, the amine group, is going to have some basic properties. Phosphates are really important to a lot of different things, and we'll learn about a lot of different phosphate things in the course of the year. A methylated compound, methyl group, is a nonpolar group. A lot of these other ones are polar in one way or another, okay? But the methyl group is a nonpolar, and again, that's going to have an effect on the function of the molecule that it's attached to, or the part of the molecule that it's attached to. So it's important to be able to identify what those are in terms of the structural formulas and know what properties they impart to the molecules they're attached to. This is some basic vocabulary about polymers and monomers, okay? You should have learned this in freshman biology, okay? But a polymer, remember, is a long molecule that's made of a lot of different smaller parts, okay? The smaller parts are called monomers, and they're joined together by a particular kind of reaction called dehydration reaction. We'll talk about that in just a minute. And then they're broken apart by hydrolysis reactions, and those are really important terms. We'll notice that both of them have hydro in them, and so the hydro part should remind you that it's something talking about water. Okay, and so we're gonna look on the next slide. We're gonna watch a little video real quick that just goes into some basics about dehydration and hydrolysis and the enzymes involved for those.
Most biological molecules are very large and are built by assembling small molecules, or monomers, into long chains. The resulting molecules are called macromolecules, or polymers. A process of linking monomers, called dehydration synthesis, involves the removal of 2 hydrogen atoms and 1 oxygen atom to form water. One way this might happen is diagrammed here, where several generic monomers are shown with OH groups that could be used for linking. The animation illustrates the joining together of monomers by dehydration synthesis, catalyzed by a polymerase enzyme, and the reverse process, in which added water results in hydrolysis, catalyzed by a hydrolase enzyme. Most biological...
So, dehydration reactions, again, you're going to remove a hydrogen from one end of one molecule, whether it's a polymer or another monomer, and a hydroxyl group from the other side of another molecule. Those two parts will go together, those two functional groups will go together to make a water molecule, leaving an unbonded area here that can be combined together to make a polymer. Whether it's going from two monomers to a dipeptide or a disaccharide or something like that, or whether it's going into a very long chain to make something like cellulose or glycogen, it doesn't make any difference. The same kind of reaction occurs there. And remember, the dehydration, if you think about what dehydration means to you physically, okay, it means that you're losing water. D means remove, and the hydro part refers to water, and so you're removing water to put things together. Okay, that's what it's called dehydration synthesis a lot of times. Okay, hydrolysis is the opposite. Hydro means water, the lysis means splitting apart, so we're going to add water back in to split things apart. Same kind of thing goes here. We're going to put in a water molecule. The hydrogen part is going to attach to one unbonded area. The bond is going to break here. We'll attach the hydrogen here and the hydroxyl group over here to the other one, and that's the process of breaking down molecules, breaking down organic molecules, and that happens in all different kinds of things. This is basically what happens in digestion. When you're digesting food, you're hydrolyzing or breaking down those complex molecules, the proteins and the carbohydrates and so forth that you eat, breaking them down into their monomers that can then be absorbed by your body and transported through your blood to the cells, where they can be used to make new molecules that are needed by your body. There are thousands of different kinds of macromolecules, okay? Thousands and thousands of different kinds. So we're not going to talk about every single kind, okay? But we're all going to talk about the 4 different categories of organic molecules: the carbohydrates, lipids, proteins, and nucleic acids. We're going to have 2 separate little videos, one about carbohydrates and lipids, one about proteins and nucleic acids, to talk about the properties of those and kind of the parts of them and how they're put together. But this is the end of the video, just about the basic overall overview of carbon chemistry, and we'll talk to you the next time.